Welcome to another edition of CCSD Living Environment. Today we're going to talk about enzymes. And the first thing that we need to define is a catalyst. A catalyst is an inorganic or organic substance that speeds up the rate of a chemical reaction. They do not enter the reaction itself, so they can be reused. So they help the reaction take place, but they don't actually enter the reaction. They're not actually a reactant. So they can be reused afterwards because they weren't actually used up in the course of the reaction itself. So if we look at our reaction here, I have the reactants and I end up with the products. We know this from before. The catalyst, we actually write above the arrow, the indication being here that it's not a reactant and it doesn't get used up. So when we're done, that catalyst can go be used someplace else. So why do we need to talk about a catalyst if we're talking about enzymes? Well, because enzymes are organic catalysts. Now remember, organic meant that it contained carbon and hydrogen both. In the case of an enzyme, enzymes fall under the category of proteins. Remember, proteins were one of the organic compounds. Most enzyme names end in ase. And if you remember, we said sugar names end in ose. Well, enzyme names end in ase. So many of the enzymes that we're going to refer to end in ase. And if it gives you a name and you're not really sure what it is, if it ends in ase, you can bet it's probably an enzyme. Enzymes are very highly specific. An enzyme will only work on one reaction. So if you have a reaction where you're putting two things together, one enzyme will do that reaction. And then if you want to take those two things back apart, a different enzyme will do that part of the reaction. Even if you're using the same things, like my hands, it's two different enzymes that will do the forward or reverse reaction. So how does an enzyme work? What actually happens to allow enzyme action to take place? The first thing that we need to do, and if you look in your note packet, underneath where you're writing this stuff, it gives you a picture and it actually numbers that picture. So you can kind of follow along each step of enzyme action according to the picture in your note packet. The enzyme needs a temporary combination or to temporarily combine with the substance that it's going to affect. So the two of them need to at some point actually come together. They need to touch. The substance that the enzyme will affect is called the substrate. So the enzyme and the substrate need to temporarily come together. The second step, the enzyme has a pocket or a space where the substrate or reacting molecule can fit. That pocket is called the active site. It would make sense that where the action or activity is going to take place on the enzyme, we would call the active site. Enzymes are usually much larger than whatever substance or substrate they're going to work on. So you have your enzyme, you have your substrate. The enzyme usually is quite a bit larger. Then the association between the enzyme and the substrate we call the enzyme-substrate complex. So most of these names are relatively easy, but you have to make sure you remember them. So the substrate is what the enzyme works on. The active site is the part of the enzyme that's going to do the job. And when the enzyme and substrate come together, we call it the enzyme-substrate complex. Step number 4, when the enzyme-substrate complex is actually together, that's when enzyme action is going to take place. And really, that makes sense. If the enzyme and substrate have come together, then the action, whatever that action is, will be able to occur. When the reaction is complete, the enzyme and the products of the reaction are going to separate. The products go on to wherever they need to be, and the enzyme can then be reused to do exactly the same job again. Remember, enzymes are highly specific, so they're only going to do one job, and they'll do it over and over and over again. You can reuse an enzyme many, many times. Eventually, like with most things, they do get old, they do get worn out, so you do have to replace them every now and then. But an enzyme can do its job many, many, many times. So I tried, and we all know my art skills aren't very high, but I tried to kind of give you the same picture that's in your note packet. So I start with the enzyme. Here's my active site. Here's my substrate. You can see where this substrate is going to fit into this enzyme. Again, highly specific. That shape is highly specific. When the enzyme and the substrate are together, that is the enzyme-substrate complex. Finally, when we're all done, the enzyme and this products come away from each other. The enzyme could go meet up with that substrate again and do the same action. So that enzyme can be reused multiple times. When you go through to do this, and in my diagram, you can sort of tell, in the picture in your note package it's a little more clear, the enzyme is larger than the substrate, and more importantly, the enzyme has not been changed. Nothing has happened to the enzyme during the course of the reaction. That is a very important thing to understand. And remember, the enzyme speeds up the reaction. It makes the reaction happen faster. It makes it happen like 1,000,000 times faster. So would the reaction still occur without the enzyme? Well, yeah, maybe someday, but not nearly as quickly as it does with the enzyme there. Enzymes don't always work alone. Every now and then they need a little bit of a help. They need a little helper to kind of get them to do their job. So we have two examples of that. The first one being a coenzyme, which is a non-protein part attached to the main enzyme. Usually a coenzyme is a vitamin, and then I have a cofactor, a non-protein part attached to the main enzyme, usually a mineral. Hopefully a light just went off for you and you're thinking, wait a minute, is that why I'm supposed to eat foods that have vitamins and minerals? Or is that why I take a multivitamin every day? Yes, it is. Remember, our reactions that occur need enzymes. They happen so much faster. We're decreasing that activation energy. We're getting it done quicker. Well, for those enzymes to function, every now and then they need a helper. And those helpers are usually vitamins and minerals. And it says non-protein part. In other words, these things are not organic. So that takes us to the question of, okay, how does the enzyme and the substrate come together? How do they fit together to make this activity work? There's two theories on that. The first one being a lock and key model. Now, if you think about a padlock and the key to unlock it, when I put the key in the padlock and turn it, the padlock pops open. So which thing is changed through the course of that reaction? The padlock has changed. The key is still the same. So that makes the padlock your substrate. The key is your enzyme. I can use that key to unlock multiple padlocks. But once I've opened up that padlock, is it still in the same orientation? No. What do I need to do? I need to spin the top back around and push it back together. That, in that case, my hand would be a different enzyme doing a different function. It may be a similar thing being the lock, but I have the key or I have my hand doing different functions, highly specific. So what happens? The substrate fits exactly into the active site on the enzyme. If you've ever had two keys that look very similar, you put one in the lock, it doesn't do anything. You put the other one in the lock and it pops open. It's because it's an exact fit, it's an exact match. Kind of like a jigsaw puzzle. That would be the lock and key model. The other option is just a little bit different. That one's called the induced fit model. So the enzyme changes shape just a little tiny bit to grasp a hold of the substrate at the active site. And if you look in your note packet, it shows you a picture of that. So the enzyme's kind of open, and as the substrate comes to the enzyme just kind of grabs just a little bit. It doesn't change shape. Remember, we can't change shape. They're highly specific. But there's just a little bit of a grab, if you will. It just grasps onto it a little bit to hold it, so when the enzyme and substrate are together, the action can take place. So it's just a little bit of a hold on to that. And then once the products have been made, they let go and that enzyme goes out to do the same thing again. So there's two different theories for how they work. And they've found both of those theories are pretty accurate when it comes to the cellular level and what's happening with enzymes. They've seen both of them function. So how do enzymes really get all this done? Well, again, we've got the enzyme, we've got what the enzyme is going to work on called the substrate. They come together, the enzyme helps that reaction occur, the products go away, the enzyme can be reused. Enzymes make reactions happen faster. The energy that it takes to make the reaction occur, the enzyme decreases that energy. That's the job of the catalyst, and in this case, the organic catalyst or enzyme. So let's take a look at some environmental conditions that could affect the way an enzyme is going to work. The first of those environmental conditions is temperature. The temperature will affect enzyme action. The rate of enzyme action increases as temperature increases. That makes sense. We're looking for the enzyme molecules and the substrate molecules to collide with each other. Well, if we increase the temperature, those molecules are going to move faster. If the molecules move faster, there's a higher chance that the enzyme and the substrate are going to collide, so there's a higher chance of the reaction taking place. And the next one is the pH. The rate at which enzyme-controlled reactions occurs varies according to the pH of the environment. And remember, when we talk about pH, we're talking about if it is acidic or basic. Some enzymes require a neutral pH to function. Some enzymes require an acidic pH to function, like the enzymes in your stomach. Some enzymes even require a basic pH to function. So depending on where they need to be, if you go too far to the acid side or too far to the base side, you can affect how that enzyme is going to work. So if we talk about the best place for enzymes to function, the absolute best temperature, the absolute best pH, that's what's known as the optimum. So optimum is the temperature or pH value where the enzyme is the most effective. Where does it work the best? We may have an enzyme that can work in our body at 25 degrees Celsius. But where is that enzyme going to work the best? 37 degrees Celsius, because that's body temperature. Now, you may have enzymes in your mouth that work best at a neutral pH of 7. Our mouth has a neutral pH. But then those enzymes in your stomach work best at a pH of 2. Can the enzymes from your stomach work at a pH of 7? Well, maybe a tiny little bit, but not very much. Why? Because their optimum pH is that pH of 2. So I sketched out the graphs that you have in your notes just so we could talk about them for a minute. In this case, I have the temperature and I have enzyme activity. So when you look at this, 40, it's saying, is as high as we want to go. And look what happens. It increases, 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 and once it hits that point, it drops right straight off. That's the optimum temperature for enzyme activity. Anything after that, they're no good. And you say, well, yeah, well, it's getting lower on this side too. True, but remember, the colder it is, well, cold isn't always so bad. They don't move around much, but on this side, they move around so much that something will happen to them. Over here, again, here's my pH, here's my enzyme activity. If this is my optimum pH, now notice I didn't put numbers on. I left that open to make a point. If my optimum pH is 7, if I move toward the acid or toward the base, it's going to affect my enzyme. But let's say my optimum pH was 4. So this is 4 now. Again, if I move toward the acid or toward the base, either way, it's going to affect my enzyme. So with pH, it's sensitive toward the acid or toward the base. With temperature, it's really only sensitive when you heat it past optimum temperature. And there's a name for that. So when you heat or when you change the pH of the environment around an enzyme and it changes the enzyme so it can no longer function, that's called denaturation. So before we get into the definition of that, I want you to think about this for a minute for me. Go back to that lock and key model. The lock is my substrate, the key is my enzyme. I can unlock as many locks as I want to, as many times as I want to with that key. But what if I took that key and I put it under very hot conditions, and I got it so it was so hot that that metal turned bright, bright red? Would that enzyme work anymore? And I hope your answer to that question is no. But what if I made it cold? What if I put the key in the freezer? Would it still work? Yeah, of course it would. It's just cold. And it may take us a little while because we want it to warm up so it doesn't stick to our hand if our hands are wet. So you want to give it a little bit of time, but as it warms up, we can use it more and more. But once it gets too hot, we can't even touch it. It melts it. It changes the shape of the key. Well, what about acid? Well, if we had a strong enough acid, we could take that metal key, put it in the acid, and guess what? It would change the shape of the teeth on that key. And remember, I can do the same thing with a strong base. Strong acid, strong base, it'll do the same thing either way. So I can make it cold, and that would be okay. But if I make it too hot, it won't work anymore. So what does the definition tell us? A change in the shape of the enzyme's active site. Remember, that's the part that's doing the job. Caused by a high temperature, strong acid or base, or a high salt concentration. A high salt concentration could also change an enzyme. If enzymes in living cells are denatured, okay, denatured, there could be a disruption to cell activity or even cell death. You would be putting homeostasis in jeopardy. So I could have a disruption to cell activity, or if it was bad enough, that cell could even die. Obviously, we don't want our enzymes to get screwed up because of temperature or pH changes, because of what that could do to the cell. So if we look at that and you think about it for a minute, so you get sick, right? And you have a bacterial infection, let's say, so you get a fever. Why do you think that happens? Well, because we're trying to make it hot and uncomfortable for the bacteria, so maybe they'll die. Well, what if the fever gets to be too high? Why do you think a high fever is scary for people? And the reason for that is because potentially you could ruin your enzymes and they could no longer allow the reactions to take place as quickly as we need them to. Same thing would be true with pH. Remember, we talked about blood pH being a very, very narrow range. If you go outside of the ranges of blood pH, again, it could be detrimental to the homeostasis of the entire organism, not just the cell. So the last couple things that I want to look at is talking about increasing the substrate concentration. So if we add more of the stuff that the enzyme is going to work to the reaction, what's going to happen? The reaction rate will increase because more substrate molecules can collide with the enzyme. Well, that makes sense. If I add more substrate, then the collisions will happen more often just because there's more of it there. So what if we go the other way? If I add more enzyme, again, the reaction rate will increase because more collisions can occur with the substrate. But eventually there comes a point where the concentration increase will not have any effect because the enzyme molecules, all the enzyme molecules that are there, have matched up to a substrate. So I don't care how much substrate you dump in, you've used up all the enzymes. They're all in a relationship at that point. So no matter how much more you add, it won't happen any faster. And so if you look in your note packet for a minute, it shows you the graph at the bottom of the page. So you can see where it increases, and then eventually it gets to a point where it can't increase anymore. And the reaction rate actually levels off. It's not saying that it's decreasing. There's nothing decreasing there. And it's not saying that it stops. But what happens is the enzyme can only work for as much enzyme as there. Once you run out of substrate, then the reaction will end. So to end the reaction, you have to make sure the substrate is gone. But as long as you have enzyme and substrate there, the reaction will continue to occur. That takes us through our conversation about enzymes. It's really important that you understand how beneficial enzymes are to chemical reactions in your cells. For us, it helps with all of respiration. For plants, it helps for photosynthesis. It helps with the replication of our DNA. It helps in the making of proteins, which, remember, enzymes are proteins. There's so many things that reactions help or are helped by because of enzymes. Those enzymes are so critically important. And remember, enzymes are proteins, they are organic. That sums up our information on enzymes for today. Have a good weekend.